questions.
We have time for some questions. Dr. Murdoch?
Okay. Bernice, I have a question on your protein diamond. What's the sensitivity of that for the telogen? And did you do?
We didn't quantify.
Okay.
The dye itself claims to have a sensitivity of 1 to 16 nanograms of phosphoprotein per band. Now, this really hasn't been done except in one other study with fish. And he did do quantification in Western blot and compared it to Western blot. And he said that he was detecting anywhere between 1 to 2 nanograms of the telogen. But he didn't give any other unit beyond that, if it was per mil, per band. So I really can't say.
I'm just wondering in these juveniles that you may be inducing it, but you may miss it in some of them due to sensitivity.
It may, and in all honesty, that probably depends on the age of the juveniles, because we did use different ages at different times. In the lowers, and especially with the ones that we were exposing to the field water, it is very possible that we weren't detecting it because it wasn't being stimulated at a high enough production level. Although you can detect it, it is reported to be detected as early as stage 58 in xenopus. This is done with relatively very highly sensitive methods, and it's always stated that it's pretty much, you can detect it at that stage, depending on your method. And it's very variable at that point. With the hepatocyte function, like I said, it's not reported to be at stage 62. And not all of my frogs, if you noted the N numbers, made it to that point. So, and also we did biasing experiments by selecting for stage 66, because that's where we expected to see it. And with the juveniles in the field, with the males, I would say we could definitely see an induction, but with the juveniles in the field, some of them were really metamorphosed, like those at Coleman Lake we collected literally as they hopped out of the pond, and we still saw a 60% reduction. But the juveniles at Snow Creek, and one of the reasons especially with the fields, I won't go so far as to say we saw an impact there. One is the low sample number of only 3. But the other is that was from a male and 2 juveniles. And the 2 juveniles were at stages, they were probably 6 to 8 months old. So it's very likely that they could be capable of producing the telogen, but they're not sexually dimorphic for me to distinguish whether or not they were male.
You said that you lost quite a few when you were bringing the field water in and putting it into the tanks.
Yes.
What do you attribute to their losses?
Okay. I should mention that, and I actually had it in here. There were 2 things. The significant mortalities occurred in 2 treatments, in Cane Creek and in controls. Now, what happened with the controls? I won't go so far as to say it was tadpole virus. I will say that there was symptomology of tadpole virus. Some other gray tree frogs had been brought in tadpole form, and we were raising them in the lab as well, and they were also showing symptoms of tadpole virus. And all of the controls that died had gross edemas. I mean, there wasn't any of them that died that didn't.
Did you do anything to the water to try to treat it, like filter it or UV light sterilization or anything like that?
No, we did not. Now, interestingly enough, the Cane Creek, the mortality that happened only happened in one replicate. During the exposure days 31 through 35, 17 out of the 20 tadpoles died. They all had no reported deformities. And I honestly can't explain that. It didn't happen in my other replicate. They actually, up until that point, seemed to be doing perfectly fine. I will say that the creek had a pesticide smell, probably the day that we did that water collection, and that may be possible. But one of the reasons we didn't do any filtration or something was try to mimic as much as we could as what they would actually be being exposed to in the field. So the best I could only control temperature to keep that from having any impacts. As once again, I've raised these several times now in the lab, and I was able to see that my temperature variations did actually induce a lot of differences in mortality. But those were the only two treatments that we had with high levels of mortality.
On the same question, what did you do to reduce the tadpole-eating disease outbreak in the controls?
Oh, we pulled them. As soon as we saw it, as soon as they had reobserved symptoms, we went ahead and pulled them and took blood from them, just to ensure that we would. I knew they were going to die, all of them would die. And I had to minimize it to where I actually did have 50% of controls left alive to test at the end of the experiment.
I have another question. It's a little different. It's since you did finally get some of the antibodies later to do the Western blots with,
Yeah.
couldn't you have gone back and taken some of the frozen samples and run more sensitive tests using those antibodies?
I have a very small container of antibodies. So I'm limited in what I can do with that.
You got a picture of
And
that
in all
Western?
honesty, this experiment was done to be a snapshot as to what we were seeing in the environment. It wasn't meant to be precise. I had to develop my methodology. I had enough work to do without. And one of the things that happened with the controls dying off, like I said, we set the endpoint for 50% of controls metamorphosing. Well, the 50% of the controls that were metamorphosing died at stage 61 to 63. Or we had to pull them at that point because when they showed tadpole demon virus. So at roughly 60 days, I would have been able to pull this exposure or this whole exposure. But with them dying off, I then had to wait for the other 50% to come up through metamorphosis, and that's one of the reasons it ran for 29 days.
One final question. This is, you didn't cover it, but I'm just curious. Do you think that increased vitellogen in the later stages is due to gene induction or increased messenger RNA stability?
In later, it's a hepatocyte function. Your hepatocytes only become competent enough to produce vitellogen at roughly stage 58, and even then you're only seeing like 1 to 2% capacity for that in the hepatocytes themselves. So xenophus, can't necessarily speak with the native endurance, but the xenophus, that's what's reported in the literature. And as they go through metamorphosis, the hepatocytes gain more competency to produce this, and that keeps increasing throughout metamorphosis and on to adulthood. Yes, ma'am.
At any time when you were exposing your frogs to the various water from the other environments, did you see any kind of odd, like behavioral changes, like anything maybe you couldn't necessarily quantify, but sluggish behavior, just odd, just clearly?
Well, no,
unorganized?
initially, when we first set this experiment up, I had to go out of town, so I collected my water, and Dr. River actually, and I had it sitting in the tanks. He actually put the tadpoles in. Initially, when you put the tadpoles into Snow Creek, they all filled the bottom of the tank. And he thought they were dead, and this experiment pretty much ended at that point. And after about 20 minutes, they were able to revive, and they went on through the metamorphosis. But beyond that, I would say in general, I would think that the Snow Creeks tend to act just a little bit more sluggish. They're not, the other tadpoles and such are just swimming constantly in there, and they're just kind of hanging out, not moving in whatever area they happen to be in. Yes, Dr. Sauter, and then I'll get you.
Okay, well, I'll have to note in addition to that, some of the experiments we've been doing with much earlier tadpoles and shorter exposure, sometimes Snow Creek ones do act very lethargic compared to controls. They just don't move around unless you disturb them. Other times they don't. So we're probably seeing a similar effect, except probably run more